Hello, myself Purvan. I am going to present part of my master's thesis titled Cost of Road Transport in an Indian Ant During Relocation. To begin with, the first three developmental stages of Holomotabolous insects, egg, larva and pupae, are together called the brood. In social insect colonies, brood ecloses into the workforce and future reproductive of the colony. Thus, survival of the brood can directly impact the fitness of the colony. But unlike other hymenopterans such as wasps or bees who abandon their brood in the old nest when relocating, ant colonies carry their brood with them while relocating to the new nest. This makes the relocation process in ant colonies complicated and riskier. In this study, we tried to estimate the cost of brood transport in ants during relocation by manipulating the amount of brood present in the colony. More specifically, we looked at immediate cost associated with relocation that can affect colonies' long-term fitness. We looked at time, if it takes more time to relocate in presence of brood, work organization, how the work organization of the individual ants is getting affected, and additional work, if the ants need to do any additional work to transport all the adults and brood items successfully. Our model organism for this study is Diacoma indicum, and 1 cm Ponerine ant. While relocating, all the adults of this species are being transported only using tandem running. During tandem running, an informed individual, the leader, form a tandem pair with a naive nestmate follower, and transports are from old to the new nest while maintaining frequent contact throughout the journey. The brood of the colony is generally carried by the followers of the tandem pair as seen in the video, or sometimes leader can also carry the brood. In total, we performed 3 sets of experiments. Colonies were kept unmanipulated during control experiments. All brood were removed 24 hours prior to the experiments in brood depleted colonies, and additional brood was added 12 hours before experiments in brood enhanced colonies. In B plus colonies, 146% additional brood was added, which was a significant amount of brood. After respective treatments, the colonies were made to relocate to a provided new nest in an experimental sand arena. The first question we asked, will they relocate at all after different treatments? As can be seen, all 12 colonies relocated in control and B-, while only 7 out of 12 colonies relocated in B+, which was significantly lower. In B+, all 12 colonies discovered the new nest. So, increasing brood makes colonies less prone to relocate. Next, what happens to the relocated colonies? To check that, we have divided total relocation time into 3 time phases. The discovery time to discover the new nest, the latency from discovery until one transport occurs, and the time it took to transport all the adults and broods are all found to be comparable. Even the number of transports performed across treatments are also comparable. So, relocation dynamics are similar across all treatments. Now, did the work organization among the leaders change? We found that work distribution among leaders was comparable in all cases. Only a few leaders did higher number of transports, while most of the leaders did only a few transports. The pattern in which relocation progressed with time did not differ across treatments, so work organization remains unaffected. But if you can remember, we have added about 146% additional brood in B+, which is a significant amount of brood. If the ants hadn't performed any additional transport or spent additional time, how did they successfully transport all the adults as well as brood? To answer that, we have divided the total transport into 3 categories: TR, when 2 adults are being transported; TRB, when the follower is holding a brood item; and BT, when the leader is transporting a brood item, and compared it across control and B+. We found that the percentage of TR significantly decreases in B+, with an associated increase in TRB and BT. Thus, additional brood is transported by increasing coupled adult brood transport and brood transport. To summarize, increasing brood makes colonies less prone to relocate. Having no brood does not increase relocation efficiency, as in no case B- colonies had any advantage over control colonies. Colonies increase coupled adult brood transport and brood transport in the presence of higher brood. The ability to seamlessly integrate brood transport with relocation will be highly favored by natural selection, especially during colonizing at new territories. In the future, we need to explore brood enhanced colonies' ability to relocate in varying conditions to understand the brood transport behavior in depth. Finally, I'd like to acknowledge Mr. Vasudev Ghosh for maintaining the colonies in the lab, Ant Lab and ISAR Kolkata for their support. Also, a big thanks to everyone for listening to my work. Thank you.